chemical engineer and packaging technologist by background, he brings over 25 years of experience in manufacturing, R&D auditing, lab testing, and consulting. Having worked with Clariant, with Ciba, Bureau Veritas, BASF, Intertech. He founded GVS Cibatech in 2015, and for the past 7 years, he has supervised over 800 projects in packaging and 100+ projects on chemical characterization. It is now over to Mr. Shivkumar Vishwanathan for Analytical Challenges in Extractable and Leachable Studies. A big round of applause, please.
Thank you, ma'am, for that kind introduction, and thank you for Spinco and Shimadzu for inviting me to speak at this MS event. And I can put this into context that we had a wonderful session in the first half talking about nitrosamines. Just imagine, whatever was talked about for nitrosamines regarding sample preparation, identification, limits, the entire list. Imagine if you had to apply all that to something that you don't know what you're searching for. And that's what is, in a nutshell, extractable leachables. Because in nitrosamine, you have the target impurity in front of you. You know what is the chemical entity that you are going after. But in an extractable leachable, you start with not knowing what you are really looking for and then going down to the specifics. So I'll slightly modify the topic from advances to analytical challenges, because if we are able to tackle these challenges, then of course, we get to actually to know what to do. You have to surmount these challenges. So, can I have the slide changer? Okay. I start off with this slide because we have talked about risk assessment in the first half, and extractable leachables also continues the same journey ahead. It is all about risk. And risk, as you know, is the product of probability into severity, any risk. And for an extractable leachable study, the severity is nothing but, is that compound toxicologically relevant as far as the patient is concerned? And second thing is the probability, like during the shelf life of the product, will this chemical entity or this compound actually show up in the drug product? So in a lot of ways, it is similar to actually nitrosamine testing and the risk assessment. Now, any good extractable leachable study should be able to, in my opinion, to be able to answer these 3 questions. One is, you have to identify, you have to have a good identification, and this could include all kinds of chemical entities and their degradation products that may come from the container closure systems, any equipments, process equipments, tubings, filters, whatever is coming in contact with either the drug substance or the drug product, whether for a short-term or for a long-term contact. Second is, you have to be able to quantify it, because these results of extractable leachable studies have to be risk assessed by a toxicologist. And unless we are able to do a good identification and quantification, the toxicologist will not be able to do the risk assessment. And this third part is not usually seen in extractable leachable studies, but as I come from a packaging technology background, it is of interest to me to know, why is this leaching happening in the first place? Is it normal? Is it abnormal? Is it expected? Is it unexpected? And if we can also try to find some of these answers early on in the drug development or the selection of the package, probably you may have a lesser challenge with extractable leachables later on. But I find this part not done to the extent that it should be. So this is basically the sum and substance of what we are trying to achieve when we are doing an extractable and leachable study. So when I generate data on extractable leachables, so extractables are nothing but compounds that are present in the packaging material and that can be forcefully extracted under exaggerated conditions. And the leachables, which should be a subset of the extractables, and these are the compounds that actually show up in the sample or in the stability sample or over the shelf life of the product. So these are some of the basic expectations. I have to find everything that is relevant. I have to identify with a good degree of confidence, and I have to quantify with a good degree of accuracy and precision. Simple and quite straightforward when we talk about the analytical methods that we use in our routine operations. This is par for the course. But this is not so easy when you talk about extractable leachables. So what are the realities? And having done hundreds of studies over so many years, we miss compounds that we probably should find. We misidentify, like something that is part of the drug product, we identify that as an extractable or a leachable, which is worse. And our semi-quantitative methods are sometimes no better than actually throwing a dart on a dartboard, because it is semi-quantitative quantitative because we start from a screening part. We basically want to try and screen as many possible targets and then move to the specifics. So these are the practical or the realities when we are attempting extractable leachable studies. Now, this is a big slide, and I purposely put all these as is, because what better forum or what better way to define this than asking the analysts themselves. So I posed this question to my analytical team: what are the challenges that you face when you are actually analyzing or when you're doing a project for extractable leachables? And some of these answers are quite specific, related to a particular method. But something that you notice here, and which I have highlighted, almost a majority of them talk about the matrix. And believe me, this is a very, very important and a crucial aspect of extractable leachable studies, because we have to work on all kinds of matrices. You have injectable products, you have oily matrices, you have a cream which contains, let's say, paraffin wax. How do you detect compounds at a ppm or a ppb level with a good degree of accuracy or good degree of confidence in such a diverse matrix? So matrix-related interference is the nightmare as far as analysts are concerned, and no wonder they have put this as their number one issue. Because if we are screening for a targeted compound, still it is doable. But if you're analyzing for a targeted compound, but when you're screening, it's quite a nightmare. So I have tried to list down the major analytical challenges that labs usually face when doing E&L studies. First and foremost is, as I said, non-targeted screening. You are looking for a needle in a haystack without knowing what the needle is. So that's what an E&L is. You have to use several techniques, orthogonal techniques. A single technique is not sufficient. So unlike any other analysis, to get to the same result of doing an extractable leachable study, multiple techniques, maybe as high as 5 or 6, need to be used parallelly to be able to do the risk assessment. Identifying the compound, I'll come to this. It's a major challenge. And a very important activity. And because it's a screening semi-quantitative study, and there is something called as an AET, which again I will describe. So, I mean, so actually the variation of the response factor in relation to the AET and how it impacts the study is something that's important. And then there are several other factors. I know that it's a huge topic and we have really limited time, so I'll try to cover as far as possible. Now, this is where we start and where we intend to finish. So the yellow oval is maybe all the extractables that are present in the container, in the rubber, in the plastic. And we use different screening techniques to try and make a list of extractables. Now, these extractables are probable leachables. They are not leachables yet because extractable study is always an exaggerated study where we use solvents, temperature, time to try and forcefully extract. And we expect that the leachables would be a subset of the extractables because leachables are those compounds that are coming out in the real life scenario. But if I get 200 extractables, can I try and develop a method or try to see whether all these 200 are coming in the drug product? That's not a very practical thing. So there is some kind of a bridging study, which can be a leachable screening or a simulation. It is known by different terms. So you try to narrow down the universe of extractables, and then you finally do the leachable study with validated methods when you have defined your targets. So this is generally how an E&L study is supposed to progress. Now, there are, as I said, complex mixtures, and you are identifying these at trace levels. So what all can these be? You have various additives, you have degradation products of these additives, you have oligomers, and then of course, nitrosamines from packaging material is also a topic. And as a part of the risk assessment, you are supposed to check if somehow nitrosamines are also getting generated from the contact materials. Then wherever you are using secondary packaging, like typically for ophthalmic formulations, you have the printing inks, adhesives, process chemicals, maybe some carryovers from the process side. So you have a very high requirement for analytics to be able to identify and quantify all these compounds. Now, this is a typical polymer manufacturing process. Now, many a times, if I get an extractable leachable project and I ask the customer, can you tell me what all is there in this rubber, or can you tell me what all is there in this plastic so that I can know and then I can sort of do my risk assessment accordingly? Many a times, not much information is forthcoming. This is simply because if you see right from the monomer, polymerization, the various steps in between, and then finally, the packaging company is converting that plastic or rubber into the product. There are so many stages where chemicals, additives, even contaminants can get introduced that it is virtually impossible to know with 100% certainty that these are all, there in my material. So that is why the supply chain is complex and lack of information can lead to surprises. Now, as was mentioned in my introduction, that chemical universe, now when we talk about non-targeted screening, it is said that the chemical universe is 10 raised to 63, which is humongous. Now, we have various databases. There are certain in-house databases, proprietary databases, public databases, which for that particular domain, they have managed to map certain chemicals or compounds of interest. These are some of the common databases. Some of you may recognize this California Proposition 65, for example, LC. LC is Extractable Leachable Safety Information Exchange, which is a not-for-profit organization, but has managed to map, I think, roughly around 600 or 650 compounds which are which are seen as extractable and leachables or which are coming actually from pharmaceutical packaging. So how do I mean, for me, getting a good database is of primary importance when I'm doing any extractable leachable studies, because I have to identify with a high degree of confidence. So this could be a way out where you know that the chemical universe is very large. I maybe try to filter it or narrow it down into that which is specifically for pharma packaging or medical devices. Within that further, I talk about elastomers. Within elastomers, again, I can maybe sub-classify it into semi-volatile. So this could be an effective way to really build compounds of interest which have a high probability of showing up in your studies, which then could make the entire process of analyzing and risk assessing that much more easier. Now, when we talk about chromatography and spectrometry, I mean, this is just like a common or actually a Venn diagram. If the gray box represents all the compounds that are there in my sample or in my packaging, out of this, which can get extracted, which are ionizable, which are separable, and out of that, which I can actually detect. So if you see, if I start with the total number of chemicals which are there in my sample or in my system, I'm able to detect only a small portion of that. So we have to be very clear that when we do an extractable leachable study, it is impossible to identify everything that is there. So you are starting itself with a certain uncertainty. And you are, I mean, based on the limitations of your analysis, the techniques that are available, you are ultimately able to identify and quantify only a limited set of compounds based on which we do the risk assessment. So my recommendation would be that we need to make a database. Current databases, I assume, focus more on the drug discovery and maybe on chemicals that are harmful, which are coming from various environmental sources. But for an extractable, leachable kind of a database, it's like very specific, related to the polymer chemistry, the rubber chemistry, and the kind of stabilizers that are used. So more work needs to be done to get a kind of a database which is relevant to E&L or extractable leachables. So we need to see what is available and we need to build on that. Second challenge, analytical toolbox. As I said, different orthogonal techniques are used to arrive at the same result. So you have, a GCMS, headspace, thermodesorption, which are all for the volatile compounds. Then you have the semi-volatiles, then the non-volatiles with LCMS, the elemental impurities with ICP, and then if you have unknowns, you need to have separate workflows to elucidate the structure. So the criteria is that all these methods, they should be exhaustive and comprehensive, but they should also be generic, and because these methods are applicable for different kinds of matrices, different kinds of packaging materials. So again, a little bit of a contradictory kind of a statement, but that's how extractable leachables are. So the methods that you have, starting from screening methods to the targeted methods, you should ensure that you are able to cover all these classes and types of compounds. Third challenge is identifying the compound using library search, and here I'm taking the GCMS for volatiles and semi-volatiles, where we have the NIST library, Wiley library, or there are other libraries and databases that are readily available. Now, there are different identification classes, and however, this is out of practical experience of doing so many studies. Many times, what we do is the compound which has the highest probability match factor, or the highest, so actually report that compound. But there is a possibility that the compound that is generating the query spectrum is not present in the library itself. Even if it is present, it's possible that there are more than one good matches. Now, when you're doing a screening, how do you then really confirm that what is getting identified or what is getting reported is indeed that compound? So the point to remember, it is not always the compound with the highest score, but you really need to look at what is it that you're extracting, what is the product, and try to look at the hits and see what it could be. I know it is not easy, but simply reporting the highest match, it can lead into errors, and we have seen that at a practical level many times. So improper use of libraries. All mass spectra should be reviewed irrespective of quality of the match factors. Do not automatically report compound that shows the highest match factor. Now, this is all very, very simple and elementary and should be done, but I'll just show this. This is a thermodesorption scan of a 3-piece ophthalmic container. So there's a bottle, which is typically a LDP bottle. You have the nozzle, which is a separate component. You have the PP cap, and then you have the label. So can you see the number of compounds that are seen? Now, how do I go back and do all this? Sorry. How do I go back and within a reasonable time do all this? So that's the challenge, that you get these kind of spectra and then you are supposed to identify your compounds of interest. Quickly, the analytical evaluation threshold, very important, where based on the daily dose of the product, you basically fix a threshold above which you will identify compounds for the risk assessment. Now, what happens is, this 80 is a calculation from the study, and then I use an internal standard at that 80 level. Now, depending upon the variation in the response, that I may be missing out if I choose the 80 wrongly. So there may be compounds that may go below, but which are, because of the response factor variation, I need to take this into account. Otherwise, I may miss compounds of interest. Just a slide here which shows that the kind of levels 0.00075 microgram per ml, how do I even detect at these levels? Especially for large volume parenterals, it's a huge challenge. Just a couple of slides on this. Is a FDA review of extractable leachable data submitted. They even want to know that you're analyzing various batches. Why is the compound increasing, decreasing? Why is it present at an earlier time point? Why it is absent at a later time point? And even going down to the recoveries of the various analytes that you have. So what I mean to say is that maybe 10 years ago, you just need to have a study done. That would be it. But nowadays, the regulators are really going through the data line by line and posing a lot of difficult questions. So unless you're analytically sound as far as the study design is concerned, it may be difficult to answer these questions later on. So I just like to end. I know that it's already past the time. For every possible extractable or leachable, there is an optimum analytical approach, and we have to find that, which will provide the most reliable identification and precise quantification. We have to invest in resources to build a platform that offers sufficient number of orthogonal and complementary approaches. Use it to document the optimum conditions and the greatest number of compounds that you can, relevant compounds that you can identify. Again, focus on the database, try to build a database, and you need to be able to constantly update this also, because it's 25 years since the FDA published its first guideline on extractable leachables, 1999. A lot of things have changed, and I foresee in the next 5 to 10 years, a lot of developments happening in this field. So this was just a quick overview of my experiences and some of the important points to note from an analytical perspective when we are doing E&L studies. Thank you. Thank you so much.